Okay, this class is about the chemical biology. This is the most students are the graduate students, and I think four of them are undergraduate students. And today class and that next class is about figuring out the size of the molecules. Also figuring out some macromolecules such as viruses that we have. Disease is so-called the COVID-19, which is a coronavirus, which you will, after this, you'll figure out the size of the molecule and particles of those organisms that we have every day. So let's think about the reminding about your knowledge about maybe you learn in this carbon and carbon structures, which is the fundamental structures of the carbon. And then you may remember or not, this the bonding length of this carbon and carbon is about 1.2 angstrom, in which we have angstrom, which we 10 angstrom is equal to 1 nanometer, which is the reason I will talk about the angstrom is this is the bond length between any atom, such as you have carbon or nitrogens or oxygen, which is 1.2 to 1.4 angstrom. But you have to think about, the angles of this between the carbon and nitrogen, such as you have carbon here and carbon here, which is 1.2 angstrom. But you have to think about the another carbon, which is another 1.2 angstrom, which is, you can think about 1.2 plus 1.2, which is 2.4, which is wrong, you know it, because we have actually the tetrahedral structures of like this, in which we have a carbon, another carbon here, we see the certain angles we have. So that if you think about some cosine of this 1.7 degree, which is sp3 orbital structures, then approximately you have a carbon and carbon here, but you think about direct distance between carbon and carbon via the carbon, this is about 2 angstrom. So this scientist in a long time ago, the reason why we determine and we define about the angstrom is actually you have so many carbons in here, but because of the angle, how many carbons in it, no matter how many carbons we have, but eventually you have 10 carbons, then instead of you have 1.2 angstrom times 10 carbon, which is 12 angstrom, which is wrong because of the angle of between the carbon. So eventually that will be about 10 angstrom, which is 1 nanometers, because of the angle. So that's why the chemists define the angstrom unit instead of the actual the nanometer unit. So for example, I will talk about, this is a chemical biology class, so we'll talk about some lipid. We have a cell, and you have cell membrane here, which is a cell membrane. And everyone knows in cell membrane, which is a lipid, and lipid is about, in which you have a particular cell, which is phospholipid. And this phospholipid structure is roughly about, you have the head group, which is oxygen and the phosphate, and then you have some carbon chains below it, right? And depending on the length of the carbon, which is carbon here 2, 3, 4, 5, 6, 7, 8, 9, and 10, typically the cells, in our cells, in your cells, then you have the bilayers of the carbon, which is a bilayer means you have one layer here, and the second layer is here. So typically the carbon length, number of the carbon is 10, so number of carbon is about 10, and you have another 2 bonding of the phosphate and oxygen and phosphate oxygen here. So about 12, 1, 2, 3, 4, 5, 6, 7, 8, and 12. So as I mentioned before, about this length of this molecule is probably quite exact. We can expect that we have 12 angstrom, which is 1.2 nanometers, which is the single layers of the lipid. So we have a bilayer of the phospholipid, which is times 2, is about, then times 2 here is about 2.4 nanometer, which is also 24 angstrom. This is about the size of, I should say, thickness of the cell membrane. And then, so the proteins that you remember about the proteins that integrated into the membrane of the cell, that in the schematic figures, you have a cell membrane, for example, membrane proteins are look like this, or ion channels, so potassium ion channel, sodium ion channel, calcium ion channels are, that flow of the ions are such as calcium, so it goes in or out, that is abundant in neuron cells. It look like this. So this is across to the bilayer membrane. So the size of the protein, the means immediately suggesting that is membrane protein. Membrane protein size, their size is actually bigger than this number here, which is a bilayer membrane. So we can expect that this is actually should be more than 2.4 nanometer. Otherwise, these channels are smaller than the lipid bilayer of the cells. They cannot penetrating, these ions are going in, back and out through the cell. So that has to be larger than 2.4 nanometers. So now we'll talk about the protein, the size of the protein, how we calculate and approximate the estimated size of the protein. So then protein is consisted of what? Amino acid, right? Amino acid. So the way of calculation is how we calculate the amino acid size. Then we simply multiply it. And then multiply this as a unit that is folded to make a structures, right? So you can think about the structures. So the amino acid is, we have a 20 different amino acid. Which is, we start from the alanine, glycine, lysine, cysteine, methionine, whatever you learn, histidine. So this class will, maybe before the midterm, we will a lot of talk about the protein. So I want you to recall, which is memorize this, all the 3-letter characters of the amino acid. Also should be, there is a 1-letter system, which is glycine is G, lysine is K, cysteine is C, methionine is M, histidine is H. So 1-letter system has to be also remembered. So when in the midterm class, you have, say, methionine, glycine, alanine, lysine, histidine, tryptophan, and we will have the chemistry on this peptide, what is the product of, such as reactions of blah, blah, blah. So I want you to memorize this 1-letter system of amino acid. Okay, so we talk the size, so we go back to the original topic. So I will make a structures out of it. So you have a carbon here, the amino acid, this is amino acid structure. So you have a hydrogen, you have now a carboxyl group, and then you have the nitrogen, main group, and then you have R. So as you know, that depending on R, if this is a H, then it's alanine, CH3 is glycine, CH2, CH2, whatever, and then NH2 is a lysine, cysteine is SH, whatever you learn. Now I'm recalling your memory, the biochemistry class. So the size now. The size is when the proteins are consist of amino acid via what? Via amide bond. Right? So the amide bond, you have, say, this is the first amino acid. So this is indicating R1. And then your second amino acid is coming. So second amino acid, you have, say, I'll draw the same structures. We have NH2, and carbon, we have H, We have OH. I'll do the second. So R2. That can be random. That can be glycine, lysine, whatever structures depending on what comes in. And then we have dehydration reactions by the enzyme in the ribosome in a cell. That the dehydration reaction you get out of the water, H2O, and then you gotta finally you have a nitrogen, and then H here. I will draw a little bit longer. So this is the amide bond, right? This is amide CONH. This is amide bond. So there is an enzyme, right? When you make a synthesis of the protein, you have a tRNA comes. So tRNA comes here, and tRNA have the end of the tRNA, you have one single amino acid. This is a ribosome, looks like a snowman. And then you have a recognizing RNA sequence. The amino acid comes here. Second tRNA comes here. That has a second amino acid. They have an enzyme that connected to two different amino acids. That's how we make a protein inside the cell. So then this is kind of a simplified reaction steps that describing actual situations. So the length and size from here to here. So we have a 2 amino acid. So calculating the number of the bonds. 1, 2, 3, 4, 5, 6. So the single amino acid has 3 bonds. 2 amino acid 6 bonds. 3 amino acid 9 bonds. As I mentioned before, 1 bond is approximately 1 angstrom. Right? 1 bond is 1 angstrom. So we have 6 bonds, means from here to here, probably you have a 6 angstrom. So this, I will give you another one. Say we have, say, a 0. Say we can imagine that we have another amino acid before the R1. Before the R1, we have a C alpha and then we have a NH2. And then this R0. So that simply we have add 3 bonds, 1, 2, and 3 bonds as the tRNA adding additional amino acid. So you have the 3 bonds adding to it. So you have 9 bonds means that you have 9 angstrom here. Okay, so let's now think about this is the size how we calculate the amino acid lengths, peptide lengths I should say. Then we think about the protein scale. As the proteins consist of amino acid sequence. So the protein, say the representative protein is a lysozyme, which is lysozyme in our tongues. In our mouths, we have a lysozyme. We eat rice. There's a polysaccharide here, and then they chopped up and goes in here. They further degradations. So this lysozyme is a degradation enzyme in our mouths. This lysozyme, say, has about, I'm not sure the exact number of the amino acid, but lysozyme roughly 120. I think 127 amino acid they have. So you can think about it. Amino acid lysozyme is, say, amino acid number one. Then the second amino acid comes and goes in repeating this process. And then the end of the amino acid, it would be amino acid number 127. So remember, each amino acid bonded 3 angstrom. So 1 amino acid bond is about 3 angstrom because 3 bonding system, NCC, that I drew it before. So you have to think about 127 amino acid. So then you have a 127 amino acid times each amino acid has 3 bonds. So 3, then calculate this. About, we have 127 times 3, that is 381. So 381 angstrom, roughly speaking. Only when these amino acids are stretched fully. there's no structures. The amino acids are situations like you have eat the meat proteins and goes in the stomach, and stomachs are strong and they denature proteins and then single strand. Something like situations you have this angstrom, and then this nanometer is about 380, about 40 nanometers. But what did I say? What did I say? In previously, we have a bilayer membrane in a cell. This is bilayer membrane. Membrane the cell. This is a cell. I talk about the proteins integrated into the cell membrane. So that this is the length is about 2.4 nanometer of the bilayer membrane. So a little bit larger than 2.4 nanometer. And here is already about 40 nanometers. It's much larger than this calculations, right? So this, because this is, we have all stretched number of the amino acid. Then this is a first structure, primary structures of the protein. Now, recall your memory that you have a secondary structures of the protein, which is alpha helix, right? Alpha helix and beta strand, in which we have, say, amino acid has a hydrogen bond connected to each other. So you have alpha helically, they form of a strand. And beta strand, you have a straight line, and there's a curve, another line comes, and they have a hydrogen bond together. So actually they folded to each other, but that means this about 40 nanometers of the full length is now folded to each other. This is how the size comes on. And they pack to each other. This protein, if you have a globular protein, they have a sphere, I would say the sphere of the protein. The sphere has all the amino acid comes to each other. There's no water in there because all the amino acids pack it to each other. So now I'll say about, you have, say, because of the folding that I mentioned, alpha helix, beta strand, the hydrophobic interaction, they pack it in a certain space. So now we have a 127 number of the amino acid now packed in a certain space. Can we think about this? Instead of the sphere, maybe we have this dice-like hexagonal structures. And then you have, say, how many amino acids can be consisting in X and Y and Z. So you have basically simple, this is like a puzzle. You have a unit of 127 amino acid packing to each other. So this will be overall size of the protein. Right. Lysozyme, how this is then this is all the strand they pack it to each other. This length will be you grab this N terminus, you grab this the other end of the C terminus, you strand it to each other. Right? But actually it's not, they pack it to each other. So you think about it, I mean, it's just very simple mass, 127 cube root of 227, that is about the number that you got. Right? So that would be, there'll be X and Y and Z. They'll be packed to each other. So maybe we can calculate, say you have maybe you have a 6 amino acid packed to each other. So I will multiply 6 times 6. Then if I do 6 times 6 times 6, I got number 216, which is a little over than this, right? I will 4, I will recalculate it. Then it's 64. So this is in between. So probably this one is 5 times 5 times 5 is about 125. So it's approximately, that means we have, you can imagine situations that have 5 amino acids packed, 5 amino acids pack, 5 amino acids pack. In this space, you have all 5 amino acids packed to each other. Then probably you have 125 amino acid. That is with the length of the X and Y of the 5 amino acid. That 125 amino acids packed to each other, packed in there. So now we can very simplify. We can very simplify that the length of the 5 amino acid, because you have now lysozyme 127 amino acid, but in this cube, the 27 amino acid is packed. Then you have a 5 amino acid at each axis of X and Y and Z that are packed successfully. So then 5 amino acid length is, you have the 3 bond in 1 amino acid. So 3 bond times about 15 angstrom, which is 1.5 nanometer. So now we have the calculations over. So you have about 1.5 nanometer, 1.5 nanometer, 1.5 nanometers amino acid of the cube, which is a protein that is a lysozyme that has 127 amino acid. Right? You take, so another example, take your blood. Take your blood. In your blood, you have the most abundant protein is called the albumin. Every day we eat, almost every day. You got an egg, you have a chicken, egg, take it out, you have a yellowish and egg white. The egg white, most of the egg white is like albumin, the chicken albumin. This albumin has about 600 amino acid. 600 amino acid. Let's calculate the 600 amino acid. The size, thinking the same approximations using this cube system. Then the simple question is how many amino acids packing in X and Y and Z, so that we can all together 600 amino acids go into in this cube. Right. So I'll do the recalculating. Say I will do maybe 8 times 8 times 8. Maybe 8 times 8 times 8, then is about, if you think about this 8 amino acid times 8 amino acid times 8, we give 512 amino acid. It's a little bit less than, but it's about the same, right? I will do maybe 9 times 9. Or do again. 9 times 9 times 9, 9 times 9 times 9, 7 to 9. So it's in between. So we'll do about 8.5 angstrom each, 8.5 amino acid in this situations here. So the albumin size is about each axis of X and Y and Z consisting approximately 8.5 amino acids. To pack all the 600 amino acids go into this space. So then we have 8.5 amino acid times 3 bonds. Then you got about 25.5 angstrom, which is 2.5 or 2.6 nanometer. Now we got 2.6 nanometers now. I mean, think about it. Think about, again, the lipid membrane in the cells, the thickness about 2.4 nanometer. Now this albumin is about the same size of our cell of the membrane. Right? So that means in your textbook you saw before in your undergraduate biochemistry class. You have a cell membrane which is 2.4 nanometer, and your channels of whatever this protein integrate into the membrane a little bit bigger than 2.4 nanometers. Then you have this albumin case about 600 amino acid size of the protein. Now it's about the same size, about the thickness of the membrane. Right. Little bit bigger. to 2.6. So it means it's the proteins that is integrated into the membrane. Probably you have more than 600 nanometers. But actually, they looks like not a cube like this ideal structures, rather than they have looks like a little bit anisotropic structures that have a certain axis is larger than the other axis, right? So look like this. So probably this protein, about this number of amino acids, or a little bit less, or a little bit more number of amino acids, that is already enough to have this bilayer membrane of the cell, which has a thickness of 2.4 nanometers, then a little bit larger than this. Okay, so this is how we calculate the protein size. So this is the end of the class. We will continue the second discussion about the estimation of the molecules, which we didn't talk about the DNA size, we didn't talk about the RNA size. Also, we such as virus. The virus has a sphere structures in which you have RNA or DNA is inside, and then the proteins are covered on the surface of the outer membrane of the virus. So that since we talk about today size of the protein calculations, so next class is about DNA RNA size estimations, and we combine this together so that we can estimate now the virus size. So that we think about the size of the virus particle, which is also useful because we always think about these days about the mask and then pore size of the mask and then how the virus is penetrating through or not, going into your mouth or nose and goes into infections. So next class is about DNA, RNA, also the virus, and then we will have about the third one, which is the end of the size discussion. It's about the protein and polymer, which is the electrophoresis case. Then you have a proteins in a polymer gels, and it goes through this, and then size separation, so-called electrophoresis cases. So that is the end of the size discussion in this class. Okay, have a nice weekend. Thank you.